meeting. Magon will share a few slides in a moment, and I will first do a few introductory slides, and then we'll do a quick round of introduction so that everybody knows who is in the meeting. It'll be very short, just your name, your organization, and your role in the organization. We'll just start with a few introduction slides now. So sorry. So just a few slides to introduce this meeting, and then we'll go for a short round. So let's see the second slide. Yes. So welcome everybody. This is the Promote Project meeting. We'll start with the introduction and then a content round. Then we'll have questions. If you have questions during the presentations, just interrupt, and we will also have some room for some questions afterwards. Yeah, and then shortly, so briefly, Wageningen University and Research. For those who do not know, these are two entities actually. We have the university, which is which has about almost 3,000 employees and a lot of students. And we have the research, which is a contract research organizations, or which are several contract research organizations. So there's a lot of overlap, but at the university part, of course, we have the fundamental and strategic research, which has as an output a lot of scientific publications. And for the research part, we have more applied and pre-competitive research. And the output there is mostly patents and licenses, although a lot of our scientists are also involved in publications, of course. So just as a brief intro of our organization. Yes, next slide. So then we'll just do a brief introduction. My name is Mark Bouwens. I'm the group lead, the research manager of the Food, Health and Consumer Research Group. And I will just facilitate this meeting. So the really important people are around me, so they will give you an introduction to yourselves just now.
I will shortly stop sharing so we can also see each other.
Very good.
Yeah. So start off. Hi, I'm Madelon Dokterberg. I'm working within the Nutrition Quality and Health Group within WFBR, and I'm focusing on studying the in vitro digestion and fermentation of fiber, protein, lipids, minerals, and so on. And I have a background in fiber characterization and their impact on gut microbiota and immunity.
I go next. My name is Paul Vos, and I'm the expertise leader of the Nutrition Quality and Health Group, where we focus a lot on in vitro research in relation to gut digestion, fermentation, gut immune interaction, etc. I'm a biologist by background, specialized in immunology, but have worked on a broad range of topics, and I'm particularly interested in the combination of health and sustainability, which I think also in this topic combines very nicely.
Yes, hello. I am Josela Meijering. I work at Wageningen University at the Division of Human Nutrition and Health. And I'm assistant professor there, and my expertise is mostly on the gut, and then on gut hormone signaling. So I look at, for instance, the hormone GLP-1, which is well known in relation to diabetes, but also I look also or have a lot of expertise in the immune system. And I working on my main expertise is also the organoid models.
Okay, then I take over. My name is Gijs van Engerven. I'm a scientist and project leader working in the Biorefinery and Sustainable Value Chains group of our institute. So that's more on the non-food side, but a lot of the side streams that we are working on are actually coming from the food side. My projects mainly deal with biopolymer characterization and functionalization, and in that respect, I'm also involved in this idea, this proposal.
All right. Then let's see, George or Georg.
Yes, I'm George van Aken. I work for COSUN. There I mainly focus on the functionality of food and functional properties of food ingredients. And I've, as a hobby, more or less, I work on in silico digestion modeling.
Great, thank you. Karen, could you briefly?
Hi everyone, my name is Karin Artboge. I work for Lantmännen R&D, Central R&D. It's a Swedish-owned farmers cooperative, and I'm a project manager for food research. Nice to meet you.
Robert?
Yes, hello everyone. Robert Dixon representing Unilever. I lead the gut microbiome program here at Unilever, and yeah, my background is in gut microbiology and immunology. And nice to see you again, Madelon and Paul.
Great, thanks. I see Karen. That might have been confusing, but.
So my name is Karen. Karen Knipping. I work for Ausnutria. I'm a senior research scientist, and my expertise is immunology, microbiology, and digestion.
Thanks. Okay, now the image shifted, so there's, I have a new picture here. Peter, could you introduce yourself?
Yes, can you hear me?
Yes, very good.
Okay. My name is Peter Hendricks. I'm working for Ingredion out of Hamburg. I'm coordinating some of the activities with Wageningen and also screening through some of the ideas. And this one is probably most interesting for some of my ideas from my colleagues from the US, but it's a bit too early for them. So I'm listening in, and we'll take it from there. And good to see some familiar faces.
Great, thank you. Elaine, I see two people there.
Hi. Hi, good morning. Elaine Vaughan. In Census, I'm responsible for health science and regulatory affairs here. I have a background in dietary fibers, prebiotics, gut microbiome. And indeed, also nice to see some familiar faces, also to see Madeleine again after a while. Very nice. And I'm with my colleague.
Matthew de Rode, nice to see you. Innovation manager at Census and manager Coast RD&I. We are also a farmers cooperative from the Netherlands dealing with ingredients, and Census is indeed focusing on health promoting ingredients, and hence our interest in this project.
Great. Thank you very much. Louisa.
Yes, hello, Louisa Martin Mare, also from Lantmännen, so I work together with Karin. I'm responsible for the health and nutrition project portfolio at R&D at Lantmännen. Great to see you, new faces.
Great, thank you. Olaf.
Hello, I'm Olaf Slickers from Cordion, and I'm an innovation fellow. A background in microbiology and fermentation mostly, and yeah, we produce functional ingredients, algae ingredients, and organic acids by fermentation.
Thank you. And then Marika.
Hello everyone, also nice to see you all. So I'm Marike Schoenmaker and I'm working for Friesland Campina. And my role is in the nutrition team to support our business groups, and that means the business group ingredients, but also dairy and consumer products that we produce and sell. I have a background in biology and medical sciences, and my role within the company is also to translate science into consumer education and materials. So within that role, I'm also responsible to look into the studies that we do and that they make sense. And nice to see also familiar faces today.
Great, thank you. Annelien?
Good morning, everyone. I'm Annelien. I represent Deutsche Mills, which is a wheat milling company located in Europe. We have milling sites in Belgium and in the Netherlands. And I'm working there as innovation engineer, and I'm mainly responsible for the long-term strategic innovation projects, which are about human and planetary health.
Great. Thank you very much. I think we have everybody then, or did I miss someone? No? Great, then we can start and go into the content, you're sharing again? Right. So like I said, the scientists will introduce everything to you now. If you have any questions during the presentations, please let us know. And then, of course, afterwards, we'll also have time to answer questions and to discuss. Yes? Okay. I'd say the floor is yours.
The content. So maybe you've already seen this slide. We've also showed it during the company session or the company markets. So very brief an overview of the Promote project. So what we observe is that the health benefits of whole food plant-based diets are not leveraged fully yet to make formulated foods healthier and more sustainable. So that's why in this project, we would like to come up with optimal sources and combination of both soluble and insoluble fiber and protein to promote the gut and metabolic health. And these combinations of ingredients should give, should have optimal digestibility, have an optimal balance of health-promoting metabolites and metabolites with adverse health effects. They should stimulate a healthy intestinal immune environment. And we also would like to extend to more systemic health by looking to the gut hormone production by these fiber and protein ingredients. So why do we study protein-fiber interactions? So what's known already is that plant-based protein commonly shows somewhat lower protein digestibility than animal-based protein. So that means more proteolytic fermentation in the colon, which is associated with positive, or the metabolites are associated with positive, but also with negative health benefits. But still, we see that plant-based whole foods are associated with positive health outcome. So both locally in the gut and systemically. And what we already seen from literature is that the presence of fiber could steer the protein metabolite production. But we've also seen from literature that the source of protein is also important because, for example, plant protein could lead to different bacterial metabolites than animal protein. So that's why in this project, we really would like to have a deep dive into these interactions between protein and fiber in the gut. And next to that, what we also see more and more on the market is products fortified with fiber. But then, of course, also an important question is, what is the impact of this fiber fortification on the protein quality and digestibility? So that's also a question which we hope to answer within this project. So then an overall overview of this project. So we already thought of the different work packages. I will put my pointer. So starting off with work package 1, so the selection of the fiber and protein sources. So what we hope to receive from you is fiber and protein ingredients, which then will be compared to plant-based whole foods, such as oats or legumes. Then in work package 2, we will perform the in vitro digestion and fermentation of these ingredients, and then of course also remove the absorbable components. And these will then be these samples will then be tested in work package 3 for their effects on the microbiota composition and metabolomics. We'll determine the effects on the epithelial barrier integrity and AHR receptors. And then also in 3.3, we will work with organoid models to determine systemic effects through gut hormones. So later on in this presentation, we will give you some more details on these specific types of analysis. And what's nice about this project is that we will not only look at ingredient X giving health benefit Y, we will also try to determine the structural characteristics responsible for the health benefits, and that we will do by characterizing the protein and fiber after digestion and fermentation, so that we're really able to determine some structure-function relationships. So all the work packages on their own, they already have will already result in valuable data. But in work package 5, we will also try to combine all data to get to these structure-function relationships. And if we find interesting complex fibers or whole foods giving interesting health effects, but we don't know yet the structural parameters which are responsible for these health benefits, we could also do some fractionation of the fibers, and then again performing digestion and fermentation with these modified fractions to really pinpoint the specific fibers responsible for the health effects. And then
Can I ask you a question?
Yeah, of course. Yeah.
We were discussing here a little bit. There appears to be a clear distinction between whole foods and ingredients. Is that the case that you want to make? The difference between whole foods and let's say ingredients that are prepped into a product that can be eaten?
Yeah. So then
okay.
I can always continue with the next slide.
Great.
Yeah. So we indeed would like to compare the benefits of whole foods with, so how can we mimic them with pure ingredients in a formulated food? So we will select.
So the underlying consumer benefit or the underlying consumer question is here. I mean, there is of course an idea and a trend of ultra processed against whole food. And this is something that you want to address here?
Yeah,
or not?
Yeah, to make processed food healthier, indeed. Yeah.
Maybe just to add quickly, of course, we do not want to shift away from recommendations that whole food should be eaten more. But we think by understanding the health benefits from those whole foods, we could still make processed foods more healthy and therefore still have a beneficial outcome overall. Also for public health considerations.
So in work package 1, we'll make a selection of these ingredients. So indeed, it will be the whole foods compared to protein and fiber ingredients alone and in combination. So we also ask you to think of, or to provide us with these ingredients. So that could be either the protein, fiber. It's really up to your portfolio, I would say. Also, if you have some waste streams which you would like to valorize, so waste streams containing both protein and fiber ingredients, we could take them along in this study.
A question from Marieke here. In terms of protein, because you started also your context with talking about plant-based proteins, is that also the line of thinking, or is animal protein also in scope?
I think we could still include it because, of course, it's also interesting to have the comparison between plant and animal. It's still relevant also for this project. So, yeah.
I think also the combination of animal proteins with specific fibers could be interesting to see whether you could still optimize for interactions.
Okay, thank you.
I also see that George.
Yes, I think that for plant-based whole foods, the complexity will be much larger than for selected pure proteins and fiber ingredients. So there will be a big task in trying to combine both and to relate both. Did you think about that? How are you going to do that?
Yeah, and that's also why we would like to do some fractionation. Probably for whole foods, it's anyway needed to separate both soluble and insoluble fiber. Maybe, Kees, can you already give some more?
Well, it's of course impossible to study all the different whole food sources that are available, but it's, I think, paramount to select relevant models per kind of class, and then see if you can determine some effects, yes or no, and if so, you can of course always dive deeper and further explore. But the idea is to first start.
For example, the protein inhibitors will have a larger effect on the study than if you're not careful. That are present in whole foods.
Mm-hmm. Yeah. We have to take that into account as well. Yeah, it's a complex matrix we're working with.
Yeah, it's not only the composition, it's also the way everything is embedded inside that matrix, and that is of course important to take into account. It's also physical constraints that need to be studied in this sense.
I think also, at least how I think about it, is that we want to take the whole foods along as mostly as a kind of reference. And I guess also we can consider during the writing phase and also during the project itself, whether we want to dive deeper and indeed maybe fractionate or reduce complexity there to make the comparison more like for like. But at least to take it as a reference on what happens with whole food combinations of protein and fiber with the increased complexity also of other components. Compare that.
Yeah, and another way is of course to make kind of reconstituted whole foods where you try to, in terms of composition, try to remake the whole food and see if you can indeed get to the same effect or not. Okay. And that means that some other effect is going on.
Yeah. Okay.
Any other questions yet on this stage? Otherwise, we will continue with the second work package.
Sorry, Madeline, maybe then you'll need to think carefully about the types of whole foods, because the types of whole foods might actually depend on the combinations or ingredients that the companies are also asking you to look at.
Yeah.
What is it indeed? What is the typical whole food when I want to eat fiber? What is the typical whole food when I want to eat protein? And those should be your reference points. And then we can build upon that with the ingredients.
Yeah,
so
because it might be, if somebody comes with wheat-based ingredient, for example, then the whole food might be more bread oriented or something, you know?
Yeah.
So that all still has to be worked out then, yeah?
And to make it even more complex, I don't like to, but of course, if you eat food, usually it's also heated or it has had a fermentation step already. That will affect also the protein and its digestibility or the interaction with the ingredients, and certainly that also is true for whole food.
Yeah, that's good. We have a separate work package on this, so we really need a lot of discussions also with you, also depending indeed on the type of ingredients you will provide us with. So it's good to then really sit together and discuss which whole foods we should select to make variable comparisons.
Yeah, and how they are treated. Because
you
usually don't eat uncooked food.
Yeah. Then we continue with work package 2. So once we made a selection of ingredients and whole foods, we'll study in vitro digestion and fermentation. So we're using the InfoJest consensus protocol for this. Then of course we also need to remove all the absorbable monomers, dimers, amino acids, peptides, which will not enter the colon. So for that, we could, for example, use dialysis. And then we end up with the digested unabsorbable fiber and protein. And then with Martijn, the group of Martijn Becker within WFBR. He works with a high throughput fermentation screening model. So it's in these well plates where online or organic acids can be analyzed, pH, ammonia production. So then we can really select the interesting protein fiber combinations, which then later on can be fermented batch-wise, because for all the follow-up experiments we require quite some material. So first screening in the high throughput model, and then the batch fermentations.
And if we talk about the context, are we talking about healthy adults, or do we also take into account athletes or people that are more, older aged adults? Because I can imagine that if we talk about fermentation of protein and fiber, that also may interact with the outcomes, right?
Yeah. So for the selection of the donors for the fecal material, indeed, it will give a different microbiota composition. So it's important to make a decision on that. But I think for now, it's maybe good to just start with healthy adults. To just select one subject, because we will do many different types of analysis. So to include both infants, adults, athletes, elderly, that will become too complex. So I think for this project, good to start off with just the healthy adults. Yeah.
Within healthy adults, I think it's also important to consider their regular diets.
Yeah.
In addition to that question, can there also be a focus on infants, or is it really only adults?
Yeah, I think just adults in this project. It depends maybe also on the partners joining the project or their interest.
I think also it should be the starting point, for instance, of this high throughput screening. I think it should be as general as possible, and then I guess we could still see if there's. But then it would be more optional or more selected if there would be room to go into more specific settings or kind of optimize it for different types of populations. But I think since it is already quite complex with all the different things that we want to address, the basis needs to be as general and simple as possible.
Yeah.
Annelien?
Well, if I understand correctly, there will be some reference materials, but it will also be possible from the companies who are part of the project to offer ingredients to be analyzed in the system. Do you have an idea how many ingredients can be investigated for each partner?
I think we could handle around 2 to 3 ingredients, but not all companies will have both protein and fiber ingredients within their portfolio. So we still have to see how to handle, how to handle with that, if we have enough companies delivering both protein and fiber or not. Otherwise, we also have to select some reference ingredients ourselves to be able to make this comparison between protein and fiber.
Yeah, although I like a lot I like a lot the in vitro digestion. It's very useful. You get a lot of insight. Of course, in reality, if you change things like protein digestibility, you'll also change a lot the physiological feedback mechanisms that regulates the gastrointestinal tracts and the gut hormones. how are you going to take that in account in some way?
I don't think I completely understand the question.
Well, I mean, if you, for example, reduce the digestibility of the proteins, then more protein will enter up into the colon and into the microbiota, which will be in effect by itself. So there's a feedback mechanism there, or a different regulation mechanism there that you cannot take into account directly through in vitro digestion. You would have to adapt that then somehow.
Yeah, that's not possible, I think. Only that we see, of course, the signals, or we see the hormones. So in that way, you know what would then happen in theory. That will already give some insights. But for the models, indeed, you're very right. That is not possible. For instance, like somatostatin also has influence on GLP-1. So the upper gut talks with the hind gut. And that will be too complicated to address that here.
Yeah, I guess we will look at a high level mechanisms of interaction. So, for instance, how protein and fiber interact directly during the digestion in the gut and small intestine, gut mostly, and there will be a lot of complexity that we will not be able to mimic in vitro, which is always the limitation of in vitro approaches.
We have two more hands. Karen first.
Thank you. Maybe I missed that, but what model are you using for an in vitro digestion? Is it InfoGest or some other protocol?
The InfoGest protocol, indeed. Yeah.
Thank you.
Okay. Louisa?
Yeah, just actually then on to that, when it comes to the transit time, will that be measured? Because obviously I'm thinking the connection to desired and undesired fermentation will maybe also be influenced by the transit time.
We're planning to take samples during the fermentation so that we also, so that we're not just looking at endpoint and starting point. So that's also the, let's say after a 6-12 hour fermentation, that we also can analyze these samples on the epithelial barrier integrity. So in that way we can, study the dynamics in the gut. Yeah.
All right, thanks.
Yeah. Okay, on to work package 3. So the microbiota composition and metabolomics. This we're planning to do by 16S RNA on species levels, and then the targeted metabolomics will be outsourced. And then we will also try to really relate the data on composition and metabolomics using bioinformatic tools to really get some more insights on these, the relation between microbiota and host health. And then for work package 3.2.
Yep, what we will do here is try to have relatively simple screening assays or in vitro assays to assess what overall effects on gut epithelial barrier function are, and also how at least one relevant immune mechanism is affected through the aryl hydrocarbon receptor, because this is well described in literature that for tryptophan metabolites, for instance, this receptor is activated, and this is generally seen as a beneficial mechanism to stimulate. So what we plan to do is, of course, have the previous steps with the digestion and the fermentation. And then there will be some processing needed to have those products, exposed to the cells. Then we will focus on co-cultures of KCO2 cells and HT29 cells, which form a barrier, also have some mucus production, and there to test for either barrier degrading effects, which of course we want to avoid, and potentially also look at the challenge of the barrier and look at potential rescue effects. I guess there's also details still to work out how much we want to do there. And then have relatively simple readouts like the TEER, so the electrical resistance and transport of marker molecules, potentially mucus production. And for the aryl hydrocarbon receptor, I have a very simple screening method with a reporter cell line, and to just look at the activation of this receptor. So not go into a lot of detail, because otherwise these will all be subjects for a project in itself. So we really want to take this as relatively simple screening outcomes.
Work package.
Yes. So here we will test actually the promising compounds from the CACO system and also the cell line. We will test them in human intestinal organoids. So recently in my team we have developed a new model, and that is the apical out model. So the normal organoid models, they actually are healthy gut tissue, but they have the disadvantage that they are inside out, so to say. So you stimulate from the inner side. And now we recently have this model up and running, and there you stimulate from the luminal side. And with this model, we like to look at the secretion of important gut hormones. So for instance, GLP-1, it's quite well known now in relation to diabetes. So many of you probably know the drug Ozempic. And but actually much less is known in relation to nutrition. And I think it's very interesting here to study that. So GLP-1 is not only has not only influence on insulin sensitivity, but also on transit time. And also on food intake, because it also on gastric emptying. So in that way, it tells a lot. Besides that, we can also do RNA-seq analysis of the organoid tissue, and in that way we can actually see, the full genome changes also of the full genome, which will then also uncover underlying pathways that are changed, and which can also show maybe things we didn't know on forehand. But it can also address, like for instance, if the AHR cell line has a very interesting candidate, we might also focus then there on these effects. So depending also what comes out in the earlier stage, we can here also focus more on these pathways or mechanisms that are of interest.
Then continuing with work package 4. So we're good to stress again that in this project we'll focus both on a soluble and insoluble fiber, because whole foods are also rich in insoluble fiber, and they're more resistant to digestion, so they will also be enriched in the colon. And what we see is that they are actually commonly ignored in studies because they're more complex to characterize. It's more challenging to handle, but still very interesting to dive deeper into also these insoluble fibers, because not that much is known yet. Here you can see an overview of the type of analysis we will perform. Of course, it depends on the starting material. So first we will fully characterize the starting materials, and that will also determine the analysis, which will then be done on the digesta and fermentation samples. But we will anyway determine the amino acids, digestibility, carbohydrate content, monosaccharide composition, and the molecular weight distribution. And then within WFBR, we also have a lot of experience with lignin characterization. So that can also be included. And then in work package 5, the data will be integrated. So data of all different work packages to determine structure function relationships. And then in work package 6, we will disseminate our findings in scientific articles. So that's the overview of the project. So if there are already Any other questions on the content, feel free to ask, and otherwise we will continue with some more general information in the TKI projects.
Yeah. So are there some more questions? Some more in Elaine?
Yeah, just a general question. Obviously, depending on the companies, you'll have to make some choices on specific ingredients and fibers and proteins, and then the natural foods. So will it be possible to sort of go in a bit deeper for those particular ingredients then to make like manuscripts, for example, you know, rather than sort of a very top line comparison of everything?
Yes, I think that's definitely possible. So we also want to have one PhD project in this project. So of course, then there will be a number of publications from this project.
Thank you.
Peter?
Yeah, thanks. Just a general observation to me, at the moment, this still comes across like a brainstorm session. Yeah, you're exploring potential things that you could put into the equation, depending on which company is interested, etc. And then, okay, there's a moment where you will have to narrow down and then come to something where everyone agrees this is going to make a difference. So how will you do that? Because, you know, the complexity, I mean, George and a few more people talked about the complexity in this thing. Yeah. How can you keep it narrow enough that there's really, let's say, an outcome that is also beneficial to the partners?
Yeah, maybe that's also echoing your words, Peter. That's also a bit my concern. And also going back to the original context, right? That we want to identify optimal combinations. I think it might help if the underlying hypothesis is a bit further narrowed, because indeed it's quite an exercise. And I also wonder a bit, somehow you want to steer towards something, right? I wonder if it's not too broad as it is currently, but maybe you already have also thoughts on this.
Yeah, also in relation to that, I think if you start studying whole foods, you have to make either a very good selection of which type of whole food will tell you most. That is more, can be generalized, and maybe it's better to focus on relatively simple systems, maybe whole foods also, that are with results that are hard, scientifically well proven, that can be translated to other types of foods, rather than trying to keep it very broad and basically keep the answers relatively vague.
Yeah, good comments. Thanks a lot. Yeah,
maybe at least also to comment how, well, we or at least I see the approach. So it's a fair comment that we need to identify relevant and representative whole foods. I guess with the essays that we have selected, in my mind, what we would do is generate data with those whole foods to look at both the fiber and the protein compartment, and then the metabolites that occur during the fermentation, and the effects that we find, also more the functional effects in the cell-based assays, and to basically to observe what happens there and compare that to the separate ingredients. So that is, in my mind, the practical approach that we take, and where we expect, based on I guess literature findings and the overall higher level health effects, that those whole food compartments will generate quite different results than the separate, for instance, maybe animal protein and fibers that have so far mostly been tested and reported on in literature. I'm not sure whether that.
And if I may, sorry, George, I'm going to interrupt you first. If I may step into your shoes and answer the question of Peter and Marike here, I think indeed this is a first probing of the industry with this proposal. And depending on our feedback and our, let's say, commitment, initial commitment, I think the project will be more, let's say, steered or more directed to a certain direction. So yes, Peter, you're absolutely right. This is a sort of a brainstorm, but I see it as more of a probing.
I will give some more info on the process in a moment, so that this also will shed some light on that part, I guess.
And in the end, it's also really, we would like to co-create as much as possible the project with you, which I think is always, I'd say, it's always a bit of balance between trying to be as concrete as possible, but also still open for suggestions and making it as relevant as possible to the partners that would like to join.
We'll get to that in a moment when I'll tell something more about the process. Maybe some more content-related questions at this moment. George, you still have your hand up?
Yes, I think I was wondering how far at the end, if you find concrete results, will you also test it in vivo? Because that would be interesting, of course. That gives a real proof.
Yeah. Indeed, it would be interesting, but for this project, it's out of scope because it's
okay.
We already have we'll tackle many different angles, so it's
Yeah, but of course, this can be followed up when we find interesting or we see that some compounds are beneficial. And then later on, follow-up projects can definitely involve human intervention studies.
Yeah, or can you adhere to already existing reports literature?
Sorry?
Well, maybe some work has already been done in vivo.
Yes, definitely, and also that we will definitely look at, so that we start with materials or foods that already have been proven effects in vivo. But actually, I think the combination here between fiber and protein, that's not so much looked at yet. You see that they mainly focus only on fiber or only on protein, and then the quality of protein, but this combination, that's really new, and actually that's really important because in our whole food, what we eat, counts all there. But therefore, it's also better to start in vitro or ex vivo models, to get leads to what are the most promising compounds there.
More questions? Shall we go through the process slides just to give you a bit more detail on that? So
I had maybe it goes here into this one. The question I had was on because you mentioned a PhD. So my assumption is that this project would run for 4 years then, or what's the idea?
Yeah.
So I think you will explain that in the general, so we just
so let's
do that.
Let's go through this slide. So basically, this is a private public-private partnership project, which means that the TKA subsidy would be filling 50% of the finance, and the rest will come from industrial partners. This will be in cash and in kind. So the ratio between the in cash and kind must be at least 1:1, but the in cash ratio, if possible, may be higher. Per partner, we depending on the number of partners, of course, we will have a range between 20 and 50K for 1.5 million euros project. And partners can co-develop, of course, a detailed project. That is what we just discussed. The meetings during project, we will find representative from all partners, and they will be involved in discussion of the major parts, the experimental design, the content, and of course the results. During the execution of the project, steering board will be in place, and there must be a representative from each partner, including one representative of the work. And of course, if you have if you are in this project, you immediately have right to apply the non-protectable foreground of their field, and you have the first right to license any protected IP which comes out of this project. So those are the important boundaries here. And then this is an overview of the budget estimate. So we will go for a total of 2 million euros and a 4-year trajectory. We will then have a 50% subsidy, so that's 1 million, and the in-kind contribution will be half of that, so 500k and 500k cash contribution. If we have 5 partners, each partner will contribute about 35,000 in cash, about 15,000 in kind per year. So times 4 comes to that amount. That's for now. And then very important, the timeline and the commitments that we need there. So the call is now open. So April to June, we've said, and it's very important, the first part, to express your interest in the coming months. So for the coming weeks, I will send out the presentation and the recordings to you all, also the people that were not able to be present here in this meeting. And then it's very important to, as soon as possible, give your interest or your indication if you would like to be involved, because then we have to co-create the research proposal in June, from June till August, by all interested parties. So it's very important there that this process is done with all of you. And then the full proposal should be submitted at the 1st of September, and that's a very important moment, because when you are in there, then you actually give a firm commitment towards this project. Yeah, so the first co-creation period, you of course have full options to brainstorm and deal with all the things that that show your interest, but you really have to give a firm commitment by the 1st of September. If the subsidy is granted, we will have it by November, and then there's a contracting process from November to January. And then if the contract will be signed, that will be a legally binding commitment, and that will be envisioned in March 2025. And of course, early 2025, we'll have the project kick off then. Any questions regarding this timeline and the process here? Yeah, because then I would say if there are no further questions, then we come to I think, the end of this meeting. Yes, I will send around, like I said, the presentation and the recording of this meeting. Please let us know if you have interest in helping us with co-creating this project, because that is the most important next step that we are going through now. If there are any additional questions, you can always contact me, mainly around the process, of course. If there are any questions regarding content directly, of course, Paul and Madelon can be reached for specific content questions. You can also reach me for that, and then I will relay your questions, of course, to the experts. Any additional questions or remarks? Nice, thanks, Karin. Any questions from this team towards the potential parties?
Yeah, I have a question, because you all came up that it's a bit unclear. So with which whole foods and which ingredients we will work in this project. So is this now clear how we will continue with that, or maybe we should address that a bit, how we think? We make this more clear and also interact with you. What are your interests?
Maybe if I can add to that, maybe it would be most interesting, maybe not in the meeting now, but also when you express your interest, to express what kind of ingredients you would like to work with. The more we know on that end, I guess the more specific we can be in the proposal, or the more concrete discussions and follow-up discussions we can have.
Good point.
Yeah, I think in general to narrow down at some points and to make good choices is important. So I think that will build a project basically.
Yeah, I think also the rationale of the biological output, right?
Yeah.
Make it a bit more concise in what to achieve.
Yeah.
Yeah,
Thanks a lot. It's yeah.
Yeah, it's a great pitch. Thanks. Yeah. For us, if our colleagues in the US would show keen interest, then they would definitely be want to be involved in some follow-up discussion. It would be great if that could happen then in the afternoon, because they only start working at 2 o'clock in the afternoon. Yeah. I'm not a subject matter expert. Yeah, I could not dive into the detail. I really need my colleagues to do that.
Thanks. Good remark. We'll take it into account. Good one. Any additional questions or remarks? Then I would really like to thank everybody, and we will be in touch. Thank you for joining and all your.
Enjoy your day.
Bye. Thank you. Thank you.
Bye.
Bye.
Time for a coffee.
Yeah. Bye.